Hello everyone, my name is Bhavesh Khatri. I am here to talk about our entry for the Inspiring Science Award 2023 titled A Dimeric Proteomimetic Design Prevents SARS-CoV-2 Infection by Dimerizing the Spike Protein. The study was carried out in Molecular Biophysics Unit at the Indian Institute of Science, Bangalore. SARS-CoV-2, which emerged in late 2019, caused global COVID-19 outbreak with roughly 650 million infections and 6.5 million deaths by 2022. The spike protein of SARS-CoV-2 binds to a receptor human ACE2 via its receptor binding domain, which plays a crucial role in viral infectivity and pathogenesis. Therefore, preventing virus attachment to host cell is the most effective way to prevent virus entry into humans. With advances in protein structure determination, a structure for the RBD-ACE2 complex was reported in just a few months. The structure provided a basis for developing inhibitors targeting this crucial interaction. Professor David Baker and colleagues from University of Washington used computational de novo protein design strategies to engineer ACE2 competitive RBD binders. This is the cryo-EM structure of the designed 3-helix bundle 56-mer mini protein bound to RBD. By analyzing the cryo-EM structure, we found helix 1 and 2 contributes to the binding, whereas helix 3 stabilizes the 3-helix bundle by packing against the other 2 helix. Our idea was to design a smaller functional mimic as we truncated helix 3. Upon truncation, we noted a significant difference in stability and around 24 loss in binding affinity, even though all the amino acids involved in binding are retained. So we decided to engineer a loop connecting helix 1 and helix 2. Considering that it is a short peptide, a conformationally constrained loop should ensure the proximity of helix 1 and 2 for optimal norm into hole packing. By optimizing the loop residues, we could drastically improve the helicity, stability, and solubility of the helix helping peptide. Further, loop engineering would facilitate dimerization of the peptide with exposed hydrophobic phase-to-phase packing, as confirmed by analytical ultracertification. The beauty of this dimeric design is that both exposed sites engage in binding to two receptor binding domains, as well as triggering dimerization of the spike protein of SARS-CoV-2. We confirmed the dimerization through cryolaptor microscopy in collaboration with Dr. Somnath Datta at IIC Bangalore. The spike protein is a homotrimer, and our design peptides simultaneously engage all three RBDs of either spike protein that induce head-to-head dimerization. We also investigated the efficacy of the peptide dimer versus monomer for preventing viral infection. We designed the monomeric peptide by substituting hydrophobic residues on the exposed phase with charged residues. As compared with the monomer, the dimeric peptide demonstrated a 45-fold higher neutralization capacity, which emphasized the importance of dimeric proteomimetic design for virus neutralization. It is very exciting. However, the most crucial requirement for the development of antivirus is its neutralization potential in vivo. To address this, we used the Syrian hamster as a model for SARS-CoV-2 infection and perform a single-dose prophylactic experiment. Virus-infected hamsters experience significant weight loss within the second day of infection, whereas treatment with peptide 8 hours before infection protected hamsters from severe weight loss. Hamsters were sacrificed after 4 days post-infection, and the virus load was measured by RT-PCR. We noted a substantially reduced viral load in peptide-treated groups, and it was further validated by histopathology of the lung tissues. The virus challenge hamsters had a large percentage area of lungs affected than the peptide-treated group. Overall, this data demonstrate that our designed dimeric peptide could significantly reduce the severity of infection. The designed dimeric helix helping peptide is remarkably stable and tolerant to multiple amino acid substitutions, hence it can be further engineered to target emerging variants of SARS-CoV-2. This is the outcome of the work published in Nature Chemical Biology. Please refer if you would like more information. With that, I would like to thank my supervisor, collaborators, and everyone who contributed to it. Thank you so much TNQ and the jury members for choosing our paper as the best published scientific paper from India in life science.